In this quick video, we're gonna talk briefly about penicillin as a mechanism of action. By the way, the illustration in this video is taken from my MedS medical pharmacology course. In the description box of this video, there is a link, and that link is for the illustration. You can download it if you want it for sure. Let's jump straight to penicillin as a mechanism of action. First of all, let me introduce you the penicillin as a structure. Penicillin as a structure is made of special four membrane called beta-lactam. This beta-lactam is fused to another ring called thiazolidine. These together, the beta-lactam and the thiazolidine, named by 6-amino-penicilloic acid. That's so important to know us first. So what we got here is our beautiful bacteria. To be more specific, is a quarter bacteria, not whole bacteria. So this blue something is the nucleus of your bacteria, and it's surrounded by the cytoplasm, which is the fluid that fills the intracellular space in any cell you get. Then it's surrounded by a layer, this layer called cell membrane, as human cells got the same thing. As here or till now is the same structure as a human: nucleus, cytoplasm, then cell membrane. But bacteria have a special cell wall outside the cell membrane. This special cell wall called by peptidoglycan. Peptido means peptide. Peptide means amino acid. A chain of amino acid combines together, they made a peptide. And the glycan means sugar, which is NAG and NAM, N-acetyl something, N-acetyl something. We don't have to know it now. What is so important to you and you have to know in this cell wall named by peptidoglycan is this amino acid. This sweet amino acid should combine together with this amino acid. I'm saying this too much because English is not my native language. Anyhow, this amino acid should combine with this amino acid. But why? To make a rigid cell wall and make the bacteria so strong. So, what make this amino acid combine with this amino acid? You have an enzyme called transpeptidases. This enzyme is the key for combining this amino acid with this. In the normal state of bacterial cell wall synthesis, and synthesis is an important word to know. Synthesis means you can only target the active prophylactic bacteria. In case of inert bacteria, you can't even touch it. So, in antibiotics, you can only kill the active prophylactic bacteria. Now, my friend, you tell me, who made this sweet transpeptidases enzyme? What made this transpeptidases enzyme is unknown protein. This sweet protein is catalyzed the transpeptidases. So transpeptidases can combine the amino acid to the another amino acid and make a rigid cell wall. This is in the normal state of bacterial cell wall synthesis. This gorgeous protein, which is unknown, is located in here, and in here meaning in the periplasmic space. This space is located between the cell wall of the bacteria and the cell membrane of the bacteria. This protein is located in here. This protein is responsible for catalyze the transpeptidases. All the things we have discussed is in the normal state of bacterial cell wall synthesis. Now you tell me about penicillin and how could that sweet penicillin act as antimicrobial therapy against our bacteria. Penicillin has a beta-lactam ring, and this sweet little protein is also have a beta-lactam ring. When penicillin given, it's gonna bind to these proteins. So it's called penicillin binding protein. Penicillin is bonding to the penicillin binding protein, making so sense. So when penicillin binds to the penicillin binding protein, there is no more transpeptidases enzyme, and there is no more combination between this amino acid and the amino acid. What's gonna really happen in this case is the cell wall, the peptidoglycan of our bacteria is gonna crack. And when it cracks like this and the salts in your tissues, for example, is gonna enters your bacteria. The bacteria dead for our sweet bacteria, and that's how penicillin works, the mechanism of action of all penicillins and all subgroups. The natural penicillin, semi-synthetic penicillin or aminopenicillin, the anti-staphylococcal penicillin, and anti-pseudomonal penicillin. That's all the mechanism of action. Thank you for watching.